Everyone, myself Asis, PhD student at Indian Institute of Technology, Jodhpur, India. The research I am going to present today is the tool we developed recently for spot deconvolution and clustering and in spatial transcriptomics. So before deep dive into the usability of this tool, let's first understand what is the spatial transcriptomics. So in transcriptomics, there are several levels of resolution. So if we have sample of any tissue or blood, so if we do the bulk RNA-seq technique, we will get the average expression of the genes that are present in our sample. So here we will get no single cell resolution. So we can't actually know what are the cellular heterogeneity that are present in our sample. So to overcome this challenge, so then there is single cell RNA-seq technique where we isolate each cell, then we sequence the whatever the genes that are present in this single cell. So on the downside, we can't actually know the from where this single cell or the cell types that are present. So there is no spatial information that are present in the single cell RNA-seq technique. So to overcome this challenge, then there is this spatial transcriptomics where we can measure the transcriptome without losing the spatial context. So in this approach, we can explore what are the spatial patterns that are present in our sample and the how the different genes that are organized in our tissue. So there are several applications of this spatial transcriptomics technique. So the image what you are seeing is the tissue organization of the whole mouse. So what the researchers do for this sample, so they took the vertical section of the whole mouse and they sequence the they sequence spatially the whole section of the mouse. So here each color represent the one organ. So how one can get the spatial transcriptomics of the data? So there are majorly two ways by which we can get the spatial transcriptome. The first is the imaging based approach and the second is the sequencing based approach. So the imaging based approaches, so how they work? They work based on the in situ hybridization approach. So we have our the sample, so then we have to add the fluorescent label probes in our sample, then we do the imaging. So we have to do this sequentially to get the spatial transcriptome of our data. So the advantage of this technique is this is very highly resolved. So one can get up to the cellular of the or the subcellular level of resolution, but the downside is the low throughput. So we can get the transcriptome of the, let's say, from 50 to 500 number of genes. On the other side, there is this sequencing based approach. So where we have to put our sample on the top of the glass slide. So where there are capturing areas in that glass slide, and in that capturing area there are barcodes are there. So after that each barcode spots have these oligonucleotides that which will bind the RNAs that are present in our sample. So the advantage of this technique is that this is high throughput. So one can get the transcriptome level of all the genes that are present in our sample, and the resolution is the low. So this is the downside where the point you are seeing here, we call it spots. So here each spot comprised of 10 to 30 number of cells. So to know what are the different cell types that are present in this spot, we require the spot deconvolution algorithms. So in spot deconvolution, what happens? By integrating the single cell data, we can infer, because in this sequencing based approach, I already mentioned each spot comprised of multi number of cells. To know the cell type proportion for each spot, we require this spot deconvolution techniques. And the another area I like to mention in this spatial transcriptomics pipeline is the domain identification. So let's say you have your sample, so you obtain the spatial transcriptomics data. So now you want to know what are the major domains. Domains means the spots having the similar expression patterns. What are the major domains that are present in your data? We have to perform this domain identification technique. So where we have to identify the domains in your data, but there are several challenges that are present in this domain identification technique. So most of the domain identification techniques that don't use the spatial information that are present in your data. So these are the basically the extension of the single cell RNA-seq technique. And to overcome these challenges, there are several techniques that are recently developed. So they use the spatial data coming from the spatial information that are coming from the spatial data. But the major downside of these recently developed techniques are that they run extremely slow for the larger datasets. So to overcome these challenges, we build our tool called the SpatialPrompt. So what it do? So so it, so it is a spatially aware domain identification method. So the tool use the spatial information or the XYZ coordinates that are present in the spatial data, and also it perform the spot deconvolution algorithm. So it by integrating the reference single cell RNA-seq data, and also it is this is highly scalable, so which I will cover in the later slides. And in addition, also we build one single cell RNA-seq reference database to seamlessly integrate for the spot deconvolution pipeline. So this is the basically the brief overview of our tool. So basically the input will be the spatial data with X and Y coordinates, and the single cell data having the cell type annotations. So the basically what the tool does. So from the single cell data, here it is the single cell resolution, and the spatial data which is the multicellular level resolution. So first, using the state of the approach, we simulate the spatial data from the single cell RNA-seq technique. Here in the simulated data, here each spot having the multicellular resolution, and in the spatial data, using the graph neural net, we constructed this integrated spatial matrix. So where each spot will be represented by its own expression, and also the weighted neighbors expression, weighted mean expression of its neighbors. So in this way, a spot will be represented by its own expression, and after that the expression level of its the neighbors or the microenvironment. So for the domain identification, so the tool only requires the spatial data with the X and Y coordinates, and it will calculate this integrated spatial matrix. Then by using the simple clustering technique, just like k-means, we will get the spatially aware domains. For the spot deconvolution, it requires this reference scRNA-seq data. So like I mentioned, from the scRNA-seq data, we will simulate the spatial data, and this spatial data, this simulated spatial data, as we simulated this data, so this data have also the known cell type proportions. So using both of this, by using the non-negative Ries regression, we calculate this integrated simulated matrix, and on the top of this integrated simulated matrix, we will apply this KNN regressor to perform this spot deconvolution. So to test our algorithm, so these are the datasets we used. So there are total 7 datasets we used. Out of this four spatial, and the other 4 are the 5 are the single cell data we used for the spot deconvolution. So sorry, there are total 9 datasets we used, and these are the tools we benchmarked. So there are total 11 tools we benchmarked. So these are the tools for the spot deconvolution, and these are the for the domain identification. So first we test our data on the mouse cortex dataset. So what happens in the mouse cortex? There are sequential organization of the cell types that are present in the spatial data. So first there is L2 and L3 cell types that are present. After that L4, L5 and L6. Like this sequential organization of cell types that are present in the mouse cortex. So by spot deconvolution we can see, so the most of the L2 3 spots that are present in the top of the cortex, and after that we get the L4, and after this we get the highest cell type proportion of the L5, then L6, and this is oligodendrocytes. And in the domain identification we can see the clear sequential organization of the clusters that are there. Similarly, then we test the algorithm on the mouse hippocampus. So also in mouse hippocampus there are this CA1, CA2 and this Dennett gyrus organization is there, and like this we get the CA1, CA2 and the Dennett principal cells with very less false positive rate, and also the similar domains that are we get in our tool. Then we test our data on this human cortex dataset. So one of the major advantage of this dataset is this dataset have this manually annotated gold standard annotations are there. So these are the like layer 1, L2, L3, L4 and L5. So these are the gold standard manual annotations. So we test this 6 tools and our tool on this dataset, and our tool achieved the normalized mutual information or the NMI score of 0.7, which is the superior compared to the other tools. Then we test how scalable our algorithm. So for this we took a spatial data having 5,000 spots, 10,000 and 20,000 and 50,000 spots. So we can see our tool can perform domain identification of 50,000 cells below 2 minutes, where other tools are taking 10 to the power 4 seconds, like approx 2 to 3 hours. The similar results we observed in the spot deconvolution. So these are the AUROC scores that are obtained for the major cell types that are present in this human cortex cell, and here we can see there are mean AUROC of 0.72 we get in our tool compared to the other tools, and the similar benchmark results we obtain for the scalability test. One of the major disadvantages of the existing spot deconvolution algorithms are the results mostly depends on the references. So if you change the reference, the spot deconvolution results also change. So there is very less robustness on the existing tools are there. So to test the robustness of our tool, we took two different datasets, single cell RNA-seq reference datasets from two different environment and two different technology. So these are these two techniques, two datasets, and we perform this spot deconvolution on the mouse kidney datasets. So we can see these are the spatial distribution of the major 6 cell types. So we can see there are very similar results that we get using this both references. So these are basically the Pearson's correlation coefficient. So for this CD we get around the 0.9 Pearson's correlation and the 0.98, 0.95. So this results told the method is the very robust. So so n- so after that we publish our, so in addition to that, for the C for like I mentioned, for our spot deconvolution, the tool required this reference single cell RNA-seq data. So we built one database called SpatialPrompt DB, which have like the around 40 single cell RNA-seq datasets for from human and the mouse. Like you can visit the database. I will put the link. And also the tool which is implemented in the publicly, one can like the one can install the tool using this pip command. So now let's come to the conclusion part. So the tool is the very powerful tool for the spot deconvolution in the clustering, and the tool can handle the very large data sets, so which make the tool very highly scalable. So one of the major advantage of the tool is it use the spatial information. So so by this, the tool use all the information that is coming from the spatial technology. So yeah, so by this I want to conclude this presentation. So these are the some references I used for this presentation, and I want to thank the my guide Dr. Pankaj and all the lab members and the department, and also I want to thank the conference to give me this the opportunity. Thank you.